do those weird feelings that I don't normally have any. So I want to start with a question that I think a lot of us have had, especially those of us here in neuroscience that are interested in neuroscience, and that is, what makes us who we are? I think we're made up. What makes us who we are is the composite of both our genetic background, but also this continual changes that are induced by the environment and our experiences. So this ability of our brain to change as a result of our environment and our input experiences is called neuroplasticity. Neuroplasticity obviously can be a great thing. So I have a couple kids and I see it every day, all these amazing acts of neuroplasticity. But neuroplasticity also sometimes can malfunction in a way. And this type of aberrant neuroplasticity can lead to neurological disorders. So in our lab, we study a couple of neurological disorders. The one that I'm going to focus on for this presentation is epilepsy. So why focus on epilepsy? So epilepsy is the third most common chronic brain disorder, but it's the most common in children and young adults. And greater than 1% of the population at some point will have epilepsy. But more important than that, epilepsy is a little bit different than a lot of the other common chronic brain disorders, like Alzheimer's and other things. These other common neurological disorders a lot of times arise later in life, after the individual has already lived a full and productive life. Whereas epilepsy really can occur at any time. And it can really rob an individual of a lifetime, really, of promise and a lifetime of accomplishments. So it has this enormous personal and social impact. But really, there's not just one type of epilepsy. Epilepsy can arise from numerous mechanisms. We'll break it down into genetic. It can be genetic or acquired or some interplay of the two. Genetic epilepsies have been pretty well successfully studied, and usually they involve a gene sequence mutation that causes an abnormal expression of some protein, which leads to hyperexcitable brain. And a lot of them are actually quite manageable. But the most common type of epilepsy is actually acquired. We, in our lab, look at temporal lobe epilepsy, which involves the hippocampus, which is thought to be the seat of learning and memory and cognition. And it's the most common epilepsy in adults. But more importantly, it can be severe and it can be intractable to medicines. So a lot of these. So this is exactly the type of epilepsy, intractable to medicines and really severe, that really exacts a large cost on the individual and society. So temporal lobe epilepsy is not genetic. In fact, it has a couple of risk factors that contribute to it. So there's trauma, long seizures, and brain infection can lead to this temporal lobe epilepsy, but don't always. And the mechanism for what happens after these antecedents or risk factors is not really well understood. One example of what may be happening, this diagram is showing kind of what may be happening in the case of a severe head trauma. You may receive that initial insult of the head trauma, and you get this acute damage. You can tell there's damage going on. And different mechanisms in the brain after that acute damage like inflammation and the cell swelling will occur. And then you may get cell loss or cell death, and you get a circuit reorganization, which makes the brain hyperexcitable, and it will have seizures, and those seizures may cause damage, and you get this vicious cycle, and now you have epilepsy. So that's one idea of what's going on after one of these initial risk factors. But really, most epilepsy doesn't follow trauma. In fact, the most common antecedent is long seizures, or what we call status epilepticus. In fact, in animal models, we can induce long seizures and produce this phenotype of this epileptic phenotype in rats. So the question, so after these long seizures, what we want to try and understand is what's going on. And we don't see a lot of cell death after this type of insult. So the question then arises, how does the brain become epileptic after insults that have little or no cell death? So we see no clear structural changes that are happening. So the change must be in the function of the actual brain cells themselves. So we know that the genes that are expressed in the neurons are what really govern the function of the neuron. And so the question then that we need to answer is, which gene or genes are changed after these insults that make these neurons epileptic or hyperexcitable? Now, if we go back to the genetic epilepsies, we see examples of genes that are involved in those epilepsies. We can see that one class of genes really stands out as very important to the function of the neuron, especially in epilepsy. And that is ion channels. So ion channels kind of set the fundamental properties of the neuron. And when you have these malfunctions in ion channels, it can lead to hyperexcitability or different problems. So in our lab, we look at one type of ion channel. The HCN channels are made up of two different genes, mostly made up of two different genes in the brain, both the HCN1 subunit and the HCN2 subunit. We've seen that after an insult that leads to epilepsy, we see a marked reduction in the expression of the HCN1 channel. What's thought to be going on is that this changes the makeup of the HCN channels, which then is associated with hyperexcitability of that neuron and epilepsy. So here in UCI, we were the first to show that HCN1 dysregulation like this is associated with animal models of epilepsy. But since then, many different labs and many different models have shown that this dysregulation is important in temporal lobe epilepsy. So if this malregulation of HCN1 is so important, this is where my presentation or my project really starts. The question that we really want to get at is, how is this HCN1 expression repressed during epileptogenesis? So we hope that by understanding these mechanisms, it'll give us insight into epileptogenesis in general. But also we hope to use, if we find out what exactly is regulating HCN1, we hope to be able to use that almost as a hook and look at what other things that mechanism will be regulating, so other than just HCN1. Okay, so this is kind of where I started in the lab. But the first thing we did is to look at the HCN1 gene itself, at the sequence of the gene, and look for regulatory factors like repressors. So we found one sequence on the gene. Here we have the DNA wrapped around the histone. And we'll say this is the HCN1 gene. DNA wrapped around the histone, which makes up the chromatin. So we found this sequence called NRC sequence where NRSF, which is thought to be a potent repressor, was found. And we found that this sequence was highly conserved through species, indicating that it may be important in the expression or regulation of the gene. So we have a good candidate. And so this is what my presentation today is going to be about. Is this NRSF protein and how it regulates HCN1 and how it's involved in this aberrant neuroplasticity that leads to epilepsy. So I broke the presentation into 4 main points. Hopefully, these 4 points I want you to remember, if nothing else. But first, I'm going to show that NRSF does in fact regulate HCN1 after an insult that leads to epilepsy, and it does so through an actual physical binding of NRSF to the HCN1 gene. Then I'm going to show that enduring repression of HCN1, persistent repression of HCN1 expression by NRSF, involves epigenetic changes to the chromatin of that HCN1 gene. Then we're going to go beyond mechanism, and we're going to block NRSF's function after an insult that will lead to epilepsy, and show that it actually attenuates the development of epilepsy. And then we'll go beyond just HCN1 and show that the prevention of epilepsy is a result of a whole set of genes that NRSF is regulating that are critical to the development of epilepsy. So first, though, I'll give you a little background on NRSF. You finally get the name. NRSF is called Neuron Restrictive Silencer Factor. It was originally found in the original theory was that NRSF silences gene expression, and so it got the silencer in its name. And it was thought, in particular, that it silences neuronal genes. And so it was thought that NRSF would be found. It was found in most cell types, but it wasn't originally found in neurons, for instance. And it was basically a switch turning off neuronal genes. So it would be found in, say, a kidney cell, because you don't need necessarily neuronal genes in your kidney cell, so you have NRSF there. But where you don't have it, originally it was thought you don't have it in the brain. So it wasn't long, though, before NRSF actually was found in neurons. And in fact, it was found that it regulates instead of silences the expression of these neuronal genes. So it wasn't originally found probably because basal levels were reported to be pretty low in the brain. But interestingly, the area of the brain where it was found is the hippocampus. So we already have this good candidate, but if you do a literature research, this looks to be a great candidate. And even more interesting is early on, it was reported that it was increased with seizure activity, not these long seizures that we have in our models of epilepsy, but these seizure activity they already saw was increasing in our epilepsy. So we have this good candidate. Now the question is NRSF involved in this HCN1 suppression? We show that HCN1 after an insult, if we give tinic acid, which is a glutamate agonist, we can generate these long seizures, and this is a model of epilepsy. And we show that after these long seizures, that HCN1 expression is down. So we look at these same animals. We then look at the expression level of NRSF. And so we expected that the expression level of a repressor would go up, and that would be, if it was regulating ACN1, that's why it would go down. So that's what we saw. But really, at this point, we just have a correlation. Probably a lot of genes go up and a lot of genes go down after this insult. But we know that NRSF supposedly, theoretically, will bind to these. So we need to investigate or look a little deeper at just what the mechanism of NRSF is, or is thought to be at the time. So here we have this will be the ACN1 gene. The DNA wraps around the histone octamer modification. It's thought that NRSF will come in and bind that cognate sequence, and will recruit cofactors and other enzymes, which will then modify the chromatin, or modify the histones. And then the chromatin structure itself will shift into a tighter wound, more repressed state. So this is the idea of how NRSF is supposed to be working. So if this is true, then what we really need to show, beyond this correlation that we saw originally, we want to show that NRSF is actually binding to the DNA of the ACN1 gene. So not only did we show that NRSF was binding to the ACN1 gene, but we showed what we would expect with this increase after the insult that we procedured in the protein expression. We would expect that we would see an increase in binding, and that's what we showed, that NRSF was in fact binding the ACN1 gene at basal levels of NRSF, and that after that insult, the binding increased. So some may be asking, well, how did you actually show that NRSF is binding to the gene? So this was kind of originally, in the early parts of the project, a very difficult task. But basically what we did is use a method called chromatin immunoprecipitation, where we would take out the tissue and we would fix it with a dilute amount of formaldehyde. So what would happen is the formaldehyde would cross-link the NRSF protein to DNA that it was bound to. In this case, maybe ACN1 gene, or any gene that NRSF is bound to, it cross-links to. Then what we do is sonicate the chromatin, so it breaks it into little fragments. And then we can use an antibody to pull down specifically NRSF. And what we're left with are short fragments of DNA that we can analyze and ask, whether this fragment belongs to the ACN1 gene or not. And that's how we showed that ACN1, that NRSF is bound to the ACN1 gene. Okay. So at this point, we're showing its binding, and we're showing the expression levels that are changing. So I guess the idea is that it doesn't necessarily mean that NRSF is regulating the gene if it's just binding. I mean, most people would think that it definitely is. But I guess the idea that I have is that if I went out on the highway, there's a big truck coming by, and I jumped onto it and latched onto it, I probably wouldn't stop or regulate it. But I could prove that I had bound it. So what we need to show really is that it's regulated. In order to do that, we need to knock down or block NRSF from binding to the ACN1 gene and show that if we do that, we can prevent the repression of ACN1 that we see after that insult. Okay, so at this point, I'm going to give an analogy. I thought at this point I would be getting a little bit fatigued from my presentation. But also, I'm going to step back and give an analogy of the strategy that we use to knock down the function of NRSF, because it works so well. And besides the 4 main points that I went over, I want you to remember this because it may actually be useful to you and your people. So I'll give an absurd analogy, and then I'll show a schematic of what we do. So I call this my sick kid at the playground analogy. I have kids, right? And if you take your kid to the playground and there's hundreds of kids playing around, if one of those kids is sick, if my son Max doesn't know that kid, then actually I'm probably pretty safe. Max may or may not get sick. The problem, though, arises when it's maybe his friend, Taylor. They love each other. I like the little bit. If Taylor is sick at the playground and Max goes to the playground, she will find Max and get him sick. And then Aaron will get sick and I'll get sick. And I'll never graduate because I'll never be in school. So one probably inefficient and super expensive way to prevent this would be to make a whole bunch of decoy fake Maxwells. There's tons of Maxwells that we can scatter throughout the playground, and then hopefully one of those decoys would take up her time and he would be safe. So this is the strategy that we really use. We know, I already showed that NRSF is definitely binding the HCN1 gene, so we know a sequence that NRSF likes to bind to. So we can make short little fragments of DNA and flood the neurons with these fragments. And NRSF will bind to those and they act as a decoy. Or we can use these scrambled oligodeoxynucleotides or ODNs. That NRSF, like Taylor, wouldn't care about some other fake kid. Don't care about that. So that can act as a control. So what we would expect, what we're hoping, and what we would expect that if we flood the neurons, or flood the cells with the brain with these decoy ODNs, that NRSF would no longer bind as much to it. So we use that chromatin meter precipitation, and we can see that basal levels and controls after the insult that leads to epilepsy, if we use the scrambled ODNs, the non-decoy ODNs, and we see that normal large increase in binding to the HCN1 gene. But if we administer these NRSE decoy ODNs, then we aggregate a lot of this binding. So this is good, and so our strategy is working very well. So the moment of truth is then to check the HCN1 expression. We would expect that if we got this binding and NRSF was important to the regulation, we would expect to rescue that downregulation. And that's exactly what we see. So we normally see, after that insult, we normally see that repression. When we give the decoy ODNs, we see that the expression is rescued by almost normal. So we showed here that NRSF is regulating HCN1 through this physical binding. If we block that binding, we can prevent that downregulation. But we have seen in models of epilepsy that HCN1 expression is not just repressed after the insult, but it is persistently repressed after the insult. It endures for not just days or weeks, but even months. So the question then arises, what's causing this persistent repression? So I'm going to show that the enduring repression by NRSF involves epigenetic changes. to the chromosome. No one has really shown that before with regards to NRSS or to epilepsy. So what we looked at is a specific epigenetic mark or a modification to the histone that has been associated with persistent gene repression. Specifically, we looked at this H3K9 dimethylation, which is not something you necessarily have to fully remember. But the idea is just seeing that chromatin can be in this active state. And when you modify some of the histones, certain histone marks, it can be a marker of this repressed state. So we looked at 3 ion channels. We looked at HCN1. We looked at HCN2 because we're not seeing, as a control, we're not really seeing any regulation of HCN2. So we wouldn't expect to see changes to the chromatin after seizures. We don't see much change. And then we looked at Kv4.2 because Kv4.2 is an ion channel as well, and after these insults, we see a large decrease in the expression of Kv4.2, but Kv4.2 does not have an NRC. So here what we would expect to see in both of these non-NRSF and non-epilepsy regulated genes is no change, and that's what we see. But in the HCN1 gene, we see a significant increase in this H3K9 dimethyl mark, which is the mark of repression. So really, what we saw, these first 2 points can be summed up as we really got at the mechanism of how HCN1 is being regulated. But one of the interesting things that we found in doing that is this strategy of how to knock down NRSF function. So the next thing I'm going to show is that if we block NRSF's function after this insult, then we can attenuate the development of it. So up to this point, we have shown NRSF regulating HCN1 expression, but we haven't really looked at whether or not NRSF was regulating its function. And so before we go into a huge epilepsy experiment, we check to make sure that, this is just a reminder, to make sure that, after this insult that leads to epilepsy, we see this reduction in H current, HCN1 current, or HCN current. And when we use the decoy ODNs, we do in fact see that we are rescuing not just the expression, but the function. So then we went ahead with the big experiment of actually monitoring the development of epilepsy. This is just basically what we did is we would implant cannula into the brain so that we could infuse these decoy ODNs right into the ventricle next to the hippocampus. Other people have shown in the past that these decoys actually penetrate into the hippocampus very well. So then after doing that surgery, we induce these long seizures, the status epilepticus, and periodically infuse these ODNs into the brain, hopefully preventing NRSF from binding and preventing the repression of HCN1. So what we see is that when we use these decoy ODNs, we do in fact see a statistically significant reduction in the cohort of animals that receive the decoys when we compare it to the animals that receive just scrambled ODNs. This is great, but then if you parse it out and look at it over time, it's even more telling. If you look at the animals that did not have the decoy ODN, you see that there's cumulative seizure probability for each group of animals. We see that these animals continue to have more and more seizures, whereas the animals that receive the decoy ODNs, the rats that receive the decoy ODNs, we have initially an amount of seizures, but they don't seem to be progressing in those development of spontaneous seizures. So here we show that by blocking NRSF function after that insult, that we're really attenuating this development of it. But if we go back to when I was talking about the background on NRSF, the thought is that NRSF regulates a whole set of neuronal genes. And we know HCN1 is important, as many labs have shown that, but we want to go beyond what NRSF is regulating, beyond HCN1. So what we did is use kind of modern miracle of technology where we could use a microarray to check 22,000 genes at a time. This way, I didn't have to do 22,000 experiments and we could graduate this century. So we start out with 22,000 genes, and we've analyzed to find all the genes that were repressed after the insult leading to epilepsy. And we did this group of 470 genes. And then after the administration of the decoy ODN, we looked for the genes in this group that were rescued. So now we have 30 genes, a small percentage of all the genes that are being repressed, which actually is kind of interesting because it's enough to attenuate the development of epilepsy. So these 30 genes must be really important. So we have this list of the genes. All of the genes have putative or theoretical NRSF binding sites, which we would expect as we're rescuing them by knocking NRSF functional. But more interestingly, they fall into these big categories of genes that are likely to be important in the fundamental function of the neurons, ion channels and cascades and enzymes that will regulate the protein. And also interestingly, a lot of these genes individually have already been studied in relation to epilepsy on their own. And so one thing they all have in common, though, is NRSF. So it's exciting to think that NRSF may be regulating this whole set of important genes in epilepsy. So, just to recap, first, we talked about mechanism, and then the next two we talked about preventing epilepsy. So we first showed that NRSF levels are augmented after this insult that leads to seizures, and we showed that it directly binds the HCN1 gene, and that by knocking out the function of NRSF, we showed that this really is important to the regulation of HCN1. And then we showed that NRSF is regulated through these epigenetic changes. So both of these are are first in the literature. No one's really shown how NRSF is working, especially in an actual in vivo brain. So this is very exciting. But then we went beyond the mechanism, and since that mechanism helped us to understand how we could block NRSF, we blocked NRSF's function and really showed that it attenuates epilepsy, and that it's affecting this group of really critically important genes to the development of epilepsy. So if we go back to when we were talking about temporal lobe epilepsy, the reason we're studying it is because it can be so severe and intractable to medicine. And so it's exciting to know that by understanding this mechanism better and by blocking NRSF function, even that we can possibly be getting close to therapeutics that may actually help to control such a devastating disease. But even if we step back from that, NRSF regulates neuronal genes even after seizure activity that doesn't lead to epilepsy. So just this understanding of the mechanisms involved in covariant neuroplasticity, or even maybe just neuroplasticity in general, may help in other neurological disorders or other fields of study. So, just before I open it up to questions, I just want to acknowledge people that have added to my neuroplasticity and made me who I am, and especially Dr. Barham and the Barham Lab over the years. And other people from the past. And I also in particular like to thank the people that worked on the project with me. Sita and Annie now are lab techs and my manager. Sita was originally a student that worked with me, and Kristen and Romella, Petrosian, and Jimmy Mundy all helped and did amazing work in the lab. Also, I think my mom is representing my parents here. She's here. Probably the only one that, well, definitely the only one that added to who I am on the genetic side. Everyone else is pretty much just neuroplasticity, I hope. And then, especially my wife, while I was here doing this research, we did some other research and had 3 kids. So it's been a crazy time and most impressive of my life. Quite amazing. And then I'd also like to thank Dr. Wood and Smith for being on my committee, and Dr. Robinson on my family. And that's just one. So with that, I will let you ask some questions. So I have to get more specific. I wanted to have this accessible and not confusing because I presented in the lab meeting.
So, I understand that the NRS is trying to certify, and that is associated with epilepsy, but then there's a leak that I don't see, because I think that you are implying that all of her genes are important, contribute to it. And couldn't they just be
yeah,
hangers on?
Yeah, so definitely they could, and in fact it would be strange if there weren't genes that were more important to epilepsy and less important. Yeah, so this is a candidate gene list, but was knocked down from 22,000 genes to 30 genes that probably have a lot more, should be investigated, but need to be investigated. Some, probably 10 on the list have been investigated in association with epilepsy, but no, you're definitely right. I don't mean to be making believe that all 30 are definitely critically involved in this, but safely.
So I believe there are several known splice isoforms to NRSF, which is also called REST. And so I was wondering if you knew whether your antibody could tell different isoforms apart, because I think some are brain-specific and others might not be.
Yeah, so there is an isoform. So NRSF is also called REST, there's a REST4 isoform that is thought to be a dominant negative. It doesn't have one of the binding sites for cofactors, and it's thought to not function. So our antibody looked at all of the NRSF splice variants that are in the brain. We have contemplated looking with different antibodies, but the one antibody that can really delineate between NRSF and that REST4, which is probably the most important. There's not a good supply of that. Middle 4 used to negative, it doesn't at some point participated.
Okay. I also have another question. So the H3K9 dimethylation, that's an interesting choice. How did you come to that one? Because most people would look at the trimethylation. And once you have that particular model of repression there, do you think something in all the cells or just some of the cells? Because the idea would be that that gene ought to be shut off. So you should see no HCM, some.
Well, we think H3K9 dimethylation because one cofactor of NRSF is the G9A enzyme, which specifically dimethylates this site, this H3K9. And from what I've read, the dimethylation is more of a repressive state, but I mean, the reason there is because we've seen that G9A. And so in vitro, in cell lines, G9A is ordered with NRSF and during this form of dimethylation in the cell line. Another lab has done that. The group are allowed. But really, we're the first to show it in vitro. So, I mean, the reason we picked that is because it's really true.
Questions from the audience? Yeah, Robinson.
So, you are a candidate for Dr. Philosophy, right? So I'll ask you a philosophical.
Very good.
So early on, you posed who we are, and you said there are genes and there are environment. Now, there are many people on the planet that also include a spiritual aspect of that. So are you negating the
well, I was going to in the intro today to kind of help you. Some people, it's our environment, our experiences that causes this change. Some people would include our choices. Some people wouldn't even include that. So some people believe we have choice and don't. I don't want to get into that necessarily in the intro, but yeah, I think that for me, spirituality would be taking a step, whether or not you have choice or no choice. So what your question is?
well, who are we? And is there a non-biological?
Is that a word? Yeah, I would think that that's not something proven.
Is that
Be testable. I mean, you would probably fail at most. I mean, you would build an experiment and it wouldn't tell you one way or the other. If someone could build an experiment and tell that there is a spiritual aspect, then I think that would be a huge deal. I should get more than a PhD.
Sean's staying in the lab to do a postdoc, and he's no longer going to be studying epigenetic. He's going to be studying hypogenetic, and that is precisely the study of how spirituality influences Other questions from the audience?
One of your last slides, you showed the time axis versus the cumulative seizure, right?
Yeah.
You don't need to show it, it's very good. So, can you explain that to us? So, is that average per animal?
Yeah.
I'm asking for two reasons. One is, so, let's say on the second day, you have 20 seizures, or what does it mean? That all the animals pulled together had 20 seizures in the second day? Or is it an average? Or what do you know?
This is the app.
So on average,
Or no, this is the total of the group. There's 6 rats. So this is the total.
so that's
the probable. Yeah.
so it means that on the second day, 6 animals, you had 20 seizures. Spontaneously.
3. I mean, that.
Oh, it was 3 in
This is, yeah. there's 6 animals in each group, but this is the total.
All right. And how you, I mean, so what was the variability? It just seemed to me, surprisingly high for the second day.
I'm not sure. None of our collaborators.
I'm going to correct somewhat your question. So, I'm sure your viewers asked this question. So, what about the, really far out time point? So, if you go out like months, do you see?
We're in the process of doing that right now. Our collaborators have to figure the EEG, and we're doing that right now. Yeah, it's important too. So we're just saying continuing. Well, probably.
Questions? Non-selective neurons, other questions? Okay, well, thanks. The next step will be a reception with all kinds of goodies as a collective. First we'll come through, and Sean will still have to stay here with his